In this lesson, we're going to look at how gene expression determines cell type through cell differentiation. So as a review, again, DNA, RNA, protein, trait. And remember, all cells in your body have the same DNA because they all started out as that single zygote. So all of your cells have the same DNA. And then also remember that environmental influences can change how genes are expressed. Okay, so that can be changing the amount of proteins, activating or deactivating genes, things like that. All right, so first let's look at just a couple different types of specialized cells. So this is a nerve cell, and remember we talked about how its form was based on its function. All right, muscle cells. Notice they are dramatically different than a nerve cell because of the different functions that they have within the body. And so, how do these cells become so different even though they have the same DNA? And the answer is simple. Every cell just doesn't use all of its DNA. It basically has all the genes to make any type of cell, and it just uses the ones that it needs. And the process by which they become different is called cell differentiation. In other words, cells become different. And what they do is they simply turn on different genes that they need to be the specific cell. So a nerve cell will turn on certain genes to become a nerve cell, and a muscle cell will turn on different genes to become a muscle cell, and that's how they become different. So as an example, hemoglobin is a protein that's only expressed in red blood cells because it is used to carry oxygen. And none of your other cells need that to happen. So even though every other cell has the gene for hemoglobin, it's only turned on in red blood cells. So the other way I kind of think about this is, remember, DNA is kind of like that cookbook, and it has all the recipes to create all cells in your body. But different cells only use different parts, and they only use some of the parts. So for example, a certain cell type may only use the parts that are highlighted in red, whereas a different cell type would use a different set of pages. So in a red blood cell, it has the genes for lactase, which is used to break down sugar, melanin, that's what colors your skin, hemoglobin, cytochrome C, and based on environmental influences, it's going to activate only the genes that it needs. So a red blood cell doesn't need lactase, it doesn't need melanin, it doesn't need cytochrome C. From this set of genes, it's only going to activate hemoglobin, which is the gene that it needs. And it's going to be able to tell which genes to turn on based on environmental influences. And most of these are signals sent by other cells telling it what it needs to be. So now let's look at stem cells. Stem cells are cells that have not differentiated. In other words, they're like blank slates. And they can come as embryonic stem cells, so when the embryo is developing, and these can become any type of cell, or they can become adult stem cells. And adult stem cells can only become certain types of cells. So once you go past the embryonic stage, you only have adult stem cells. So here's an adult stem cell, which can only become one type of cell. So for example, it could become, you have some adult stem cells that can become red blood cells, or some that can become muscle cells, or some that can become nerve cells. but they can only become that one type of cell. An embryonic stem cell, on the other hand, can become any type of cell. So it could become all three of those. And that's important because when a single cell, so you started out as a single cell and became a whole organism full of a multitude of different cells, that's because you had embryonic stem cells, which the initial one was the fertilized egg, and then it, as it split, would differentiate into all the other different types of cells. So that's how that one zygote can become a complex multicellular organism that has all kinds of different specialized cells. Those embryonic stem cells activated different genes and became those specialized cells. Now, stem cell research is something that has the potential to solve a lot of diseases. Things like if you have liver disease, you could take stem cells and actually regrow an entire liver. People that have other kind of muscular diseases, you could regrow muscle. You could regrow nerves in order to fix people that had spinal cord injuries. So there's a lot of potential for stem cells to be used for medicine. Unfortunately, they're very controversial because really you can only use embryonic stem cells for most of these things. And embryonic stem cells are usually only found in embryos. And the only way that we can get embryos are from discarded fertilized eggs. And so people associate that with abortion and other things that they may have ethical or religious issues with, and so it becomes this big thing. And so right now, stem cell research on embryonic stem cells cannot be funded by the government. However, there's a lot of hope that in the future we'll be able to actually take adult stem cells and turn them back into embryonic stem cells, and then we could do all kinds of things and potentially open up a whole new avenue of solving diseases and other issues.